welcome you all to today's webinar. Today, we will discuss about the upcoming 3 days workshop on the introduction to computational biology and biomedical data science. This workshop is designed for graduates, postgraduates, faculty, and researchers. Before we start with the talk, I would like to introduce us. We are a US based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy to use analytical tools, and our mission is to make bioinformatics more accessible. OmicsLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. I'm honored to have been working with a team that collectively has amazing experience in this area. Dr. Alfred Tauber, who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation. One of these projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panu, who is an expert bioinformatician and helped prepare many of the materials we will use. Evie Titiski, who is a computer scientist and leads the development of the T-Bioinfo platform we will use. Dr. Harpreet has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases, and looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar, who is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. Over the past several years, he has been developing collaboration with universities as a mentor and project lead for Omics Logic training and research fellowship. Dr. Raghavendran Lakshmi Narayanan is an accomplished computational biologist with experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein data structural bioinformatics. Applying his proficiency in using Python, R, and Shell scripts for bioinformatics, Dr. Raghavendran is interested in translational approaches to understand cancer biology, neurological disorders, and bioinformatics. To discuss more about the workshop, schedule, and the outcome, I would like to invite one of the co-mentor and bioinformatics specialist, Dr. Mohit Mazumdar. Dr. Mohit Mazumdar, over to you.
Thank you, Sparsh, and hello, everyone. Welcome to this session and welcome to this webinar. So my name is Mohit Mazumdar, and as many of you might be joining for the first time, so I have a PhD in computational biology, and I have years of experience working on various biological problems, doing research in bioinformatics using computational tools. So today we will start by learning why we are doing this first, and then we will learn about the various things associated with the workshop and also the schedule and the experts who will be talking in this important workshop that we will be conducting from 26th December to 20th December. So advances in sequencing and computational biology have, drastically increased our capacity to explore biology through this vast amount of data. So our understanding of the world of biology is changing due to many breakthroughs in biotechnology, in agriculture, in infectious disease research, and also in other life science research. So that is one of the key reasons that we designed this workshop for beginners and students who are starting their journey with their specializations, specialization degrees that that you are enrolled in. So students who are doing BTech, students who are doing masters, MSc, uh, MTech, PhD, and people who are doing research, especially clinicians who would want to learn about the innovative industrial technologies related to biological data analysis. So this program and this workshop, especially for anyone who would want to have that, who have this keen interest in understanding that what is going on. So in today's world, we are learning from newspapers, we are learning from different sources about these different breakthroughs that are happening. That starts from the development of vaccines to all the new advancements that are coming for diseases such as cancer and other life-threatening diseases. So, and people who are interested in doing that, people who have interest in learning about that, it is quite challenging to stay up to the speed and to learn that what is going on and what are the ways we can contribute, what are the ways we can get into it and start doing the same. So in this session, we will learn about topics and the curriculum that will be covered that we will be covering in this workshop, and that I will be covering in this session, along with the resources that I said. We have resources that utilizes this innovative Omics Logic Bioinformatics training, and that will allow students to be a part of this growing and rapidly evolving community of researchers, scientists, entrepreneurs, and industry specialists. So this Omics Logic short 3-day workshop registrations will allow the participants to access the self-learning course. As you can see in the screen, there are some of the key objectives that we have put together. So self-learning courses and projects. So these are available on our proprietary portal, Learn Omics Logic, and platform T-Bioinfo server. So anyone who registers will have access to all those resources for 30 days from the day of the registration, and a training on biological and biomedical data analysis using sequence and structural data, and then utilizing the big data which is available on public repositories. So we will do that. And over the past few years, I would like to mention this, that our innovative technology that is being utilized by multiple resources and by creating pathways for students to excel in their career has led to many awards and recognition, as you can see on the screen. So in India, we have collaborated and piloted our training programs with 30 universities for the last 3 years, testing and validating the project-based curriculum to develop research ideas and research projects, and to support this process, and to support this entire process from training to be able to participate in real and cutting-edge research. So this actually brings us to talk about this opportunity, this another opportunity for students and faculty who are interested in exploring the vast multidimensional data and to gain skill sets that are really truly relevant to industry and also academic research, and that pertains to deeper understanding of human beings and all the other living beings. So this Omics Logic workshop is designed for students and researchers were eager to kickstart their journey to big data by understanding the application from a biologist perspective, and solve the challenge like an expert in computational biology. So that's what we are going to expect from you after you go through this training. So this, especially this version of the workshop, is a special opportunity, as we are glad to announce an academic partnership with a Prema's Life Sciences. And to tell you more about this partnership that will be helpful for all of us going through this experience, because Prema's Life Sciences in India is a young, dynamic, and a focused organization introducing these game-changing niche technologies such as genomics, this application in cell biology, biopharma, to boost innovative research and diagnostics in India compared to the developing countries. And during this session, the students from the experts will learn about the cutting edge and niche life science tools for the advancements of Indian research and clinical diagnostics. So it'll be a great opportunity in that context. So in this workshop, and in order to get you all a complete understanding of big data from experts, we designed this workshops with Prema's Life Sciences that will help address the gaps. So that will be done by doing a pre-orientation workshop on 26th December on NGS data generation, and we will cover next generation machines and some of the coolest application in biomedical research. So let's learn about some of those topics that will be covering in this pre-orientation session. So this workshop will commence on 26th December, which is a Sunday. And the first keynote session will be about Illumina machines and data generation. So there will be a pre-orientation session also before that for you to get started and have all the login information and all the access and all of that. And then we will start with the session. The first keynote session will be from 10 a.m. to 11 a.m. IST on 26th December. Followed by that, there will be another session which will be about the application in clinical NGS, and we will learn about important terms and important things that are going on in clinical NGS, such as oncology testing. We will learn about rare complex diseases. We'll learn about reproductive genetic diseases and genetic health and microbial wellness and whole genome sequencing. So these are really, I mean, topics that the scientists are working on and using the insights from those sort of experiments, and technology is helping us to take critical decisions for drug development and all of that. So we will go to that one by one in this entire workshop, and really excited to learn more and more about it, and also to tell you more and more about it. So after learning about this clinical application, we will get to learn about the advanced technologies that are emerging to provide even deeper understanding of each of the cells. So in this last session of the day, Dr. Nityanand Sharma, Senior Product Manager, Single Cell and Spatial Genomics, will talk about single cell genomics and clinical paradigms. So you'll learn from data generation and then clinical application, and then you will learn about the advanced applications. So followed by this, the next part of the workshop, and the next part of the workshop, we will be learning about NGS machines. After learning about the NGS machines and clinical application, we will be learning from the industry experts about the biological data analysis, to learn about big data, to learn about databases, to learn about public repositories, and to learn about basics of data science and computational biology. So these are some of the things that will be covered in the next 2 days, and we will have experts for that. So for the next 2 days, along with me, we will have experts from our team to take you through these different topics that I will go through in a while, in some time. But before I do that, let's hear from our experts who are present in this webinar. Let me invite Dr. Raghavendran first, who is an expert mentor and a big data specialist. So over to you, Dr. Raghavendran, for the introduction.
Thank you. Thank you very much, Dr. Mohit. Let me start by sharing my screen. I have to share everything so that you can, yeah. Okay, I think I'm audible and my screen is visible. Yeah, so I'm Raghavendran. I'm a research consultant and a mentor with Pine Biotech and Omics Logic. I'm trained in bioinformatics and I hold a PhD in computational biology from the Max Planck Institute of Germany. My research interest and focus is on data science techniques such as machine learning, deep learning, and several other methods applied to the integration of multi-omics data like gene expression data, gene mutation data, and copy number variation data, etc. So my role in Pine Biotech includes developing curricula and mentoring students in the research fellowship program. So let me talk about one such example in which I developed machine learning models to predict tumor responses for a variety of treatment options, employing multi-omics integration of gene expression data with gene mutation and copy number variation data. So, excuse me, we know that cancer is the leading cause for disease deaths worldwide. So for such a dreadful disease, for a long time, our systematic cancer treatments have relied on chemotherapeutic drugs that are generally toxic for all the cells and marginally more toxic for the cancer cells. This is a problem because on an average, only 25% of the cancer patients respond positively to cancer drugs. But in comparison, about 80% of the patients respond to popular painkiller drugs that are available. So a prominent solution to such a problem is precision oncological therapy, where the treatment strategy is designed by targeting the molecular characteristics of the patient's tumor. Hence, the treatment is personalized to patients who are likely to respond to specific cancer therapy. There is not sufficiently enough data from cancer patients to molecularly characterize tumors for different multitude of treatment and other therapeutic options available. So hence, we have to resort to a suitable model system. In this particular data, the model system uses patient-derived xenografts. And what are patient-derived xenografts? The tumor cells from the cancer patients are subjected to multiple molecular profiling through next generation sequencing techniques. And this gives information about the gene expression data, gene mutation data, and epigenetic variation data that were extracted. These data molecularly characterizes the tumor, and then these tumor samples are then grafted into immunosuppressed mice, and these immunosuppressed mice, which hold several different cancers, were administered multiple treatment strategies and tumor therapeutic options, and the tumor's response to these strategy are studied in terms of volume, size, and survivability. So this data records the reaction of these tumors to different treatments. Both the molecular characterization data and the treatment response data are available along with the publication that was published in 2015. So such a wealthy of data, generally, it generates a lot of interesting questions. So my specific interest was to use this multitude of data to design and implement machine learning models so that I can predict tumor responses to specific different treatments. So most of the biomarkers data that are obtained to date include only one omic measurement, either genomics or transcriptomics or other omics data that are available. And thus, they do not take full advantage of the complexity that is present in the system, which are captured by measuring multi-omics data. So there are several methods as listed here to integrate several multiple omics data into one. But selecting the best appropriate method and strategy that will unravel the complementary information and integrate these omics data into a meaningful feature is still a very difficult challenge. So for this particular data, I used a very late integration of multi-omics integration, and I also tried several other methods, but this one proved very, very useful. So what happens when I do that? Traditionally, in treatment response identification research area, the prediction to any treatment, the treatment response prediction is the treatment response is predicted using the single gene biomarker that usually characterizes the tumor or onset of the oncological condition. But if this is a problem, because this single gene marker may characterize the onset of the tumor, but it might not give us enough information about the prediction of the treatment response. So that is proved here, where the MCC coefficient, which describes or which describes the accuracy of the machine learning model, really suffers to even cross 0.5. So when I use information from all the all the genetic data, then the accuracy increases marginally. So thereby, by applying 1, 2, and even 3 more omics data to the same data set, my accuracy not only increases, but also the precision also increases in terms of accuracy. So, so what could we get? Usually, what could we gain from this from such a data? There, so one major conclusion is multi-omics data definitely improves the accuracy. Another major conclusion is it's very difficult to integrate several layers of multi-omics data into a very many meaningful data that can be leveraged. So what more could be done? Such an analysis can be extended to include patient data from TCGA to just verify whether machine learning models that are trained on model data, mouse model or cell line model data, how does they perform when they are tested on the patient data? And with this machine learning model data, we can develop or we can generate a set of interesting or important features that will describe a concise list of genes or biomarkers that can be extended to gene set enrichment analysis kind of analysis steps to identify specific markers that can characterize the treatment response also. So hope this was informative and inspiring for all of you to generate interesting research questions from publicly available data. And such methods and such techniques will be briefed in this workshop that is aimed at introducing several omics data and how these omics data as independent or integrated analysis will help you understand relevant biological systems. We will demonstrate you using multiple data sets that are publicly available and are curated for demonstrating the logic behind the complex analysis pipelines involving machine learning or deep learning methods. In the course of this workshop, we will also showcase the resources we designed to help students and other users to master the necessary data science methods at 3 different levels. First level is at the logical level, which doesn't require the user to be an expert at coding, where you can leverage the key bioinfo platforms for your analysis by concentrating on understanding the logic behind several machine learning methods that are incorporated into the big data platform. And then analysis using R for those who are comfortable at R coding. Our code blocks and detailed step-by-step explanation for those codes will enable you to understand machine learning methodology using R. And then analysis using Python. For those who are interested in Python, our lessons, our lessons involving coding using popular online coding resources like Google Colab will enable you to understand, execute, and modify our codes to fit your data of interest and will help you master machine learning and deep learning employing Python libraries. I wish and I hope for very engaging participation throughout the workshop that will improve our scientific understanding in these different multitude of scientific research areas. I thank you all for your attention and will be happy to take any questions if you have any. And I hand over the podium back to Dr. Mohit. Let me stop my share and.
Okay, thank you, Dr. Raghavendran. Now, let me invite our multi-omics expert, Dr. Harpreet Kaur, who has been contributing immensely to the bioinformatics community by helping, mentoring, and publishing important resources for the researchers. Over to you, Dr. Harpreet.
Thank you, Dr. Mohit and Dr. Raghav. Now let me share my screen first. Hello, everyone. First, let me introduce about myself. So my name is Harpreet Kaur and I have a PhD in bioinformatics. In my current role, I'm curriculum developer and research consultant. So here I am mentoring research fellows as well as working with international collaborators in various research projects based on biomedical NGS data. So I would be one of the mentor in this upcoming workshop. In the upcoming workshop, you will gain the extensive data science skills, and that can be applied on multi-omics biomedical data. Once you learn those skills, next you can apply all those skills on your own research projects. So how you can apply various research skills, especially if you don't have any previous bioinformatics research background. So now let me first give you the glimpse of my research background, as well as let me give you the glimpse of the project that were completed by our research fellows who have no prior bioinformatics background. So my PhD was in the area of cancer genomics, where I have specifically in the field of liver cancer. So I have tried to address various concerns associated with the liver cancer using publicly available data and implementing various bioinformatics techniques. For example, in the first project, we use large-scale publicly available transcriptomics data to identify diagnostic biomarker panel for hepatocellular carcinoma. So in this study, we used 30 dataset which containing nearly 4,000 samples to elucidate transcriptomics biomarker panel for diagnosis and prognosis of the liver cancer, implementing various machine learning techniques. In the next project, we shifted to another primary liver malignancy, that is cholangiocarcinoma, which drives where carcinoma derived from cell lining the bile duct. So in this project, we use publicly available 9 dataset and which containing HCC, CCA, and the normal sample to scrutinize cholangiocarcinoma specific transcriptomic signatures, implementing various data mining techniques like extra tree, SVM, random forest, logistic regression. In the another project, we used multi-omics data. So here, not just a transcriptomics data, we also considered epigenomics data to identify a biomarker panel for stage classification of hepatocellular carcinoma. In this study, we taken the data from publicly available repository, which Dr. Raghav was talking about, that is TCGA, which is the Cancer Genome Atlas. So in this study, we identified 51 signatures, which include 30 RNA transcripts and 21 methylation CpG sites using various biomedical data techniques. In the last project, we just not consider transcriptomics and epigenomics layer. We also consider miRNA data. So we have tried to integrate all the 3 omics layers of the HCC patient to identify signatures that can stratify low reoccurrence risk group and high reoccurrence risk group of the HCC patients. Now let me give you the glimpse. If you don't have any background, prior background, particularly in R or Python. So as Dr. Raghav has shown you the platform, that is T-Bioinfo server, and some of the lessons that where we already have R and Python scripts. So we have the research fellows who have completed their research project based on publicly available data and implementing various tools from T-Bioinfo server itself. So we worked in various fields, including the oncology. So here we have undergraduate student, Ms. Sime Dolaner, who is from the Turkey University, completed her research project entitled Long Non-Coding RNA as Therapeutic Target in Chronic Lymphocytic Leukemia using publicly available data, implementing various bioinformatics tools from T-Bioinfo server. Then Ms. Urja Parekh has completed her project on identification of the biomarker to differentiate between neural stem cell and glioblastoma stem cells, employing publicly available data from the glioblastoma samples and using various bioinformatics techniques. Then Ms. Rutvi Vaja has completed her project in the field of neuroscience. Particularly, she worked on multiple sclerosis. She completed her project entitled In Silico Analysis of Transcriptomic Profiling and Affected Biological Pathway in Multiple Sclerosis. Now, let me also give you a glimpse how data science can be applied in the field of infectious disease. So here we have a research fellow that is Mr. Supantha Dey. He's worked particularly in the field of malaria, where he has explored publicly available transcriptomics data for the analysis of gene expression profile to study malaria vaccine dose efficacy and immune response modulation using various bioinformatics tools that are integrated on the T-Bioinfo server. So apart from transcriptomics data, so then we also worked on genomics data. So in this project, we worked with the researchers team from Noguchi Memorial Institute of Medical Research, University of Ghana, where we study the haplotype variant analysis of Plasmodium falciparum AMA1 based on the genomics data. Then apart from these fields, so our research fellows, after their learning, they also worked in the field of space omics. So here we have Mr. Subhrajit, first Miss Dilara Dikeen has completed her project using metagenomics data, where she completed her project on radiation threat to human in space as an alternative approach with probiotics using metagenomics data and various bioinformatics tools that are integrated on T-Bioinfo server. Then Mr. Subhrajit has completed his project again in the same field. Here he, instead of the metagenomics data, he explored the transcriptomics data. So where he worked on a project entitled Transcriptomics Analysis of DNA Damage Response in zebrafish embryos under the stimulated microgravity. So then we have also worked with the research fellows from India itself, particularly MIT University. So, for example, we Ms. Kanva Sharma has completed her project on bioinformatics analysis of small cell lung cancer patients with bone metastasis. Sorry. Then Ms. Akanksha Singh completed her project on in silico analysis of NGS data of glioblastoma to understand the impact of IDH1 mutation. Then Mr. Aditya Bajpai has completed his project in the field of virology, where he mainly explored transcriptomics analysis of CD4 positive T cell to recognize gene signatures regulating HIV infection. Then Ms. Shruti Goyal completed her project on transcriptomic analysis for early detection of cervical cancer. Then Mr. Hriday Thakur completed his project on the identification of biomarker for the prostate cancer. So besides PhDs, masters, as well as undergraduate students, as well as different researchers, so we have students from high school who able to complete their project and able to present their project on various conferences like Ms. Netra Srinivasan and Mr. Anders has completed their research project in the field of neuroscience. So we are going to in the upcoming workshop. So right now I'm just giving you a glimpse how the biomedical data and data science can be applied to drive biological meaning. So you can implement all these skills in the you are going to learn how we can actually implement even if we don't have any prior bioinformatics background. So my major research interest lies in the field of cancer genomics, infectious disease, neuroscience, machine learning, and biomarker discovery. And I'm looking forward to see every one of you in this workshop. If you have any question, please feel free to post your queries in the chat box. And now I would like to pass this session back to Dr. Mohit. So Dr. Mohit, over to you.
Thank you, Dr. Preet. Let me share my screen. Yes, and now we will learn about the sessions, how the second part of the sessions will be day 2 and day 3. And before that, let me just quickly tell you that what it will be about. So again, thank you, Dr. Preet. Now let's learn about the topic and let's learn about what would be the pace of this. So that in this introductory workshop, our focus will be to give you an in-depth overview of important omics technologies that is changing the way healthcare is, and this entire system is rapidly evolving. So our focus will be to making you learn about these complex technologies with several different layers, so that it becomes easy for you to learn, and you have ample amount of resources with you to take you which you can take on for the next 1 month to learn about all of this. So we will learn about, obviously, the next generation sequencing technologies and the data, the different omics layer data, and then we will also learn about how this it all integrates into drug discovery and how this is actually helping in the process of drug discovery. So we will start with genomics, like giving you an overview of genomics, that genomics is an essential subfield of bioinformatics, and it's a very important field now, which is a major force in expanding our knowledge about genetic association with with diseases and other traits. So it's an interdisciplinary field. So in such interdisciplinary field, we have an opportunity also to learn about other things. So the other things would be to learn about R and Python, how R and Python is used in genomics. What are the different technologies or different algorithms that we use in genomics to gain insights about different things, such as, multiple sequence alignment of genomes. Then we are talking about, DNA methylation factors. Then we are talking about massive databases such as GeneBank, EMBL, and SRA that allows us to do all of this research. So this will be focused on that, and this is what genomics is going to be all about in this workshop. And then the another technology that we are going to learn about. So there are three key technologies that we are going to cover in this workshop, along with chemoinformatics, is genomics, transcriptomics, and metagenomics. So transcriptomics is the process by which the information in a strand of DNA is copied into a new molecule of RNA. So this transcription is like a dynamic process, right? So the transcriptome is a snapshot of genes transcribed in a cell at a particular point of time. And this is very important to learn, that this is a complete snapshot of the entire genes at a particular point of time. So this actually enables us to gain a lot of information when we compare different stages, different conditions. So that's why we learn so much about transcriptomics, and that's why it is being used in so much in clinics and in research. So the sum total, that is what the transcriptome is all about. And then we are going to learn about different analytical steps. So how do you analyze, once you have the transcriptomic data, it's deposited in public repositories, and you can answer some good questions, some good biological research questions, like biomarkers and finding critical genes that are expressed at a certain point of time that gives you an idea about their function. So all of that can be done through transcriptomics. And what we are going to learn is that how do you do the processing? So from a raw data, so you will learn about the raw data on 26th December, and on 27th December, we will be learning about how do you process this raw data into something meaningful. So processing, exploring, and we'll learn about gene and isoforms, downstream analysis, statistical analysis, and then interpretation of that kind of data. So that would be one of the key objectives of this workshop, to get you comfortable with the visual T-Bioinfo platform, to get you comfortable with the research that can be done using such tools and algorithms. Sorry. And the next thing would be, after transcriptomics, is also to learn about metagenomics. Not after, but we are going to learn about these simultaneously in a way that it all makes sense. So in metagenomics, we would be learning about microorganisms, and then the samples that are collected from the natural environment. And we will be learning about the community structure, because in a natural environment, you'll find all kind of species richness, and then you'll be investigating their functional roles, and how these microbial communities are playing a key role in human health, human disease, and many sort of interactions that we are learning from this sort of data. So again, we will be going through some of the project examples, and I will take you in a few more slides the entire schedule of the program. So here we will be going through the gut microbiome project. We'll learn about that. We'll learn about brain access project, and then we will learn about how this is being useful for those who do not have any background of metagenomic technology, and people who do have a background can explore more further. There's like metagenomic sequencing, analysis, phyloseq, and all the biome analysis that one can do with the tools that we will be showcasing you, and with the resources, tutorials that you can take help from to do this on your own. So learning about the technologies, omics technologies, genetic data, and now you're looking for solution strategies. Now you're looking for molecules, you're looking into structure, you're into three-dimensional world. So from 3 dimension to 2 dimension, from 2 dimension, that is the sequence, to single dimension to multi-dimensional data, and from multi-dimensional data we move to 3-dimensional data, and again from 3-dimensional data we will move to again matrices. So again 2-dimensional data to do data science and AI. So this is really a fascinating journey from different type of data types that we were going to learn about in this short workshop. It is just to give you that what all can be done within this space. So one of the things that is going to be covered is protein folding structure prediction, because when you think about these important biological processes, you think about also the diseases, you think about also what is available and what is not available. For a computational biologist and for a person who does not have all the resources to do the experiments, we kind of rely on predictions, and we do predictions all the time. We humans do it all the time. So we also have built systems to do better prediction. One of that system is Google's DeepMind's AlphaFold. So we will be learning about how to run AlphaFold and what are the applications of AlphaFold, with different variants that you're learning about, how that influences the structure and the function. Now we are back to the schedule for actually the day 1 and day 2 after the orientation session on 26th. This is 27th and 28th, and we have sessions starting from 10:45 a.m. To 6 p.m. till 6 p.m. in the evening, and all of this will be recorded and will be shared for those who are missing out on any of the sessions. But it would be a great experience starting from the beginning till the end. We will start with the orientation of the program to the program. We will see if you have the learn profiles up, and we will see if you have the resources emailed to you and all of that you're prepared for the session to start and do some hands-on experience and learn from the experts. So we will start from introduction to NGS technologies, different type of technologies that I just described to you, by taking some examples and learning some of the pipelines, that what we can experience and what you can extract, and how do you run the pipeline, what are the different components there. So you're going to learn about genomics, metagenomics, and then transcriptomics with example pipelines. Then there's a lunch break. Obviously we all get tired, and after that after the lunch break we will have the session on how do you go next after you run the pipeline. How do you do with that? What are the different outputs? What are the different analysis method, and how do you interpret that? That would be for 1 hour. And after that we will have so these sessions will be divided into again 2 sessions. So once for the first session we will be learning about the mutation, we will be learning about genomic visualization, then we will be learning about the mutation and variant analysis that you can do with genomics. So here you are learning about genomics, you're learning about application of genomics. Before that, you got an overview of all the omics technology, and you learn about different pipelines that you can use to analyze such data. After that, when we are done with around 2:30 p.m. So after that, we start with our specialization tracks, which is from 2:30 p.m. to 3:30 p.m. And this is Indian Standard Time. So you can change it according to your time zone. Now we have in that session, we have application of NGS. So we will be learning about the TCGA project, and then we will be learning about some of the projects related to COVID-19 sequencing. So that is not mentioned here, and we have included that now. So wastewater management. So we will learn about 3 case studies in brief here, and then we will actually work on those case studies later in the last session. So here you will get an overview of what the case studies are all about and what are we going to do, and then we will have a tea break. So after the tea break, we will start with Cheminformatics application and case study. We will go over some of the softwares that are being used in Cheminformatics and give you an idea of how and what are the things that are that you can start with using some of the tools that are available online. Then we will start with the last session, which is a breakout session, where you can choose that which session you want to you're more interested in. So is it like machine learning and drug efficacy and learning about projects that are available applying transcriptomics analysis or genomics analysis or structural analysis. So we will have 3 rooms, any of you which you can join, and we'll be having obviously the recording, so you can learn about the different sessions later as well if you're a registered participant. In the second day, we will go through the statistical analysis, because all of these technologies are producing you a table. So that table is again like a matrix. It has rows, it has columns, and it is a very big table. So how do you interpret that table? We will apply methodologies, algorithms such as DEGs, some understand about the beta diversity, and we learn about from the structural perspective as well. So after that session, when we are well versed with what all that you can expect from this kind of data, what is the way to learn how to extract meaningful data, because one of the challenges with big data analysis and being a data scientist is that you have to analyze big data, and then you have to find critical information from a huge amount of information. So that's really a challenge, because data annotation is again a very trivial thing. So we will learn about that in the next session, that how you can explore unexplored data, how which you don't have no idea about. That is unsupervised machine learning, and then supervised machine learning. And we have an expert with us on deep learning, Dr. Raghavendran, as you have heard from him. So he will be taking us from that different types of algorithms that are used for such analysis. And then from a biological standpoint, what all of this analysis is making sense. You need to learn about that, right? You need to learn that why we are doing this, why we are using machine learning, and from what we are getting from machine learning, and why it is helpful for biology. So that is the connection to the biology, downstream analysis, pathway analysis, how it is affecting the pathways, if there are mutations, how it is affecting how it is becoming cancerous, and how it is applicable for metagenomics. And then we have an industrial panel discussion, and we have presentations and discussions where we will be telling you about the opportunities that are upcoming. So internship opportunities or job opportunities, or the things that you need to be prepared for to get a job in the industry. We will be hearing from some of the industry panels coming from Preorientation Life Sciences and Pine Biotech. And then we have the certification and Q&A and networking, where you can network with the community and discuss and do all of the interaction that can help you maybe get different opportunities that are there. So that's about the workshop, and that's about the entire curriculum about the workshop. And if you have any questions about the curriculum and all of that, I'm happy to answer to those questions. And now I will invite Ms. Sonalika, if she is around, to take you through the registration and this workshop registration fees and all of that, and also the resources that you will have. Thank you. Thank you, everyone.
Thank you so much, Dr. Mohit, for such an elaborated topic discussion. Now, before I proceed further and guide you all to get the access to the resources that we have on the portal, I'll just launch a poll quickly, and it would be nice if you all could answer the poll so that we can see from which all backgrounds do you all belong to. So my first question is, What is your educational background? Are you from a field of bioinformatics or data science or biochemistry or pharmacy or microbiology or some other field? My second question is, what is your level of education? Uh, are you a bachelorate? Have you finished your graduation or have you completed your master's or PhD or postdoc? And my third question is, what are your research interests? Machine learning, genomics, proteomics, transcriptomics, biomedical data science, epigenomics, metagenomics, or computer-aided drug discovery. This poll would be on for 30 more seconds. And thank you all for answering the questions in the poll. And those who have answered the questions in the poll, it would be nice if you could visit the learn.omicslogic.com portal on your Chrome or Safari and go along with me on creating an account on the learn.omicslogic portal and see how you can make the most of the online resources that are present on the portal. The poll would be on for a few more seconds. And in the meanwhile, let me just post the link of the portal in the chat box. Thank you so much, Ojasvi. Those who have created their account on the portal, please let me know by typing in a 1 or yes in the chat box. And others can just click on the link of the portal and create an account for yourself or sign up using your existing Google accounts. Let me do that for you. If you just click on the login button, you will be redirected to a sign up page. Here you can either create an account for yourself, that is just simply put in your mail ID and create a password for yourself, or use any of your Google accounts to sign up on the portal. Please let me know in the chat box once you have completed your sign up or if you have already signed up on the portal. So Manaswini, for a person with a background of chemistry, if you are interested in biochemistry, then definitely it's computer-aided drug designing or drug repurposing that you can proceed further with. And we will be having a session on structural biology, computational biology for the structural aspect of biology specifically. So there, I think you might find your research interest and you can work on it. Right, Dr. Mohit? Would you like to give a suggestion to Manaswini on that? Okay, now I would be ending the poll. Thank you everyone for answering the questions in the poll. Now, if you have completed your sign up, let me just show you quickly how you can update your profile and why is it important to have an updated profile on the portal. So if you click on your mail address on the menu bar that you can see on the portal on the top, you will be redirected to a profile section. It's very important that you have an updated profile because this is the way we get to know about your progress. We monitor your progress at the back end, and we do feature the best profiles with the best progress and scores on our social media platforms. And therefore, it's important to have an updated profile picture. Please make sure that your name is in title case and you have linked your social media accounts, and also add a brief bio about yourself. It could be about your educational background and your research interest. And also when you will be a part of the workshop, then you will get the starter subscription active for a month where you will get the access to all the courses on the Omics Logic portal that you can learn from. Now, if I take you through the portal and guide you how you can navigate through the portal, so you just need to click on the courses tab on the menu, and you will be redirected to a list of courses that you can see here. On the right side, you will see various tags which mentions multi-omics fields and data science. Now let's say you just want to see what all courses are present that you can learn from. So just click on course and every course will be displayed in the menu. And you can simply pick the course that you would like to learn from. Also, if you would like to go through any of the project examples, then all you need to do is just click on the projects and you will get a list of example projects where you will learn how the methodologies and the concepts that you will learn in the course could be applied to answer a real life research problem. I request all the participants to please mute themselves. And if you have any question, please put in the chat box for me to answer. Thank you. Okay, so now talking about the workshop registration. So if you just click on the programs tab on the menu on the portal, you will be redirected to a list of programs. Here you need to just simply type workshop and you will be redirected to the page of workshop on computational biology and biomedical data science. On this program page, when you'll scroll down, you'll find the schedule first of all. And the schedule is listed for the 27th and 28th of the workshop. We will be soon listing out the Prima's Life Sciences schedule as well. On the program page, you will find 3 different levels that you can pick and enroll for the workshop. The first level is the beginner's level, that is for $15. The intermediate level is for $30 and advanced is for $45. Now, what is the difference between these 3 different levels? In the first level, definitely you will get the access to all the resources on the Learn OmicsLogic portal. In the intermediate level, you will get the access to one-on-one mentor support that is missing from the beginner's level, where you will be getting the assistance only in group meetings. But in case of intermediate, you will get the one-on-one mentor support as well. And in the advanced license, you will get some extra time to work on any of the projects that you may propose during the duration of your working on any of the courses on the portal, right? So that is the difference between these 3 different levels. Many times participants get confused which level should they pick up. So if you want to work on a short project for submitting in your institutes as a report or just for your experience to get some hands-on, then an advanced license will be the best for you. If you are looking for some one-on-one mentor guidance, then an intermediate level will be the best one for you, or else the beginner level is always there that you can opt for. And once you pick any of the subscription types, you will get the access to all the courses on the OmicsLogic portal. Just the difference would be in the duration of the time period that you will be getting the access to. So that was the very simple process to get yourself registered for the workshop. I have some questions in the chat box. Let me just answer. If we are in the beginner and we finish it, we can upload it. Yes, you can definitely update your subscription level from beginners to advanced. That's an option that you can pick any time soon. Any other question? Farhat is saying, can it will be helpful for diagnostic person? Yes, definitely it will be. So let's say now when you say a diagnostic person, are you referring to clinical data or like a diagnosis of a particular a particular disorder or a condition? Am I right, Farhat? Molecular diagnostic, right? Okay, so I'll like Dr. Harpreet to take this question because she will be able to answer this question in a greater detail. Dr. Harpreet or Dr. Raghav, can you please take this question and tell Farhat how the biomedical data analysis actually help in molecular diagnosis?
I think I can do that. So,
Thank you, Dr. Raghav.
So Farhat, I need to know what do you mean by molecular diagnostics? So are you interested or are you familiar? Are you from a molecular biology background, or if I can see your are you working in molecular diagnostics company or laboratory kind of settings where your primary job or primary scientific interaction is identifying molecular diagnostics. Can you please little bit more specific? So in any case, I assume that you are going to be familiar with molecular biology. And okay, excellent. In that case, this course will actually enlighten you, on this workshop will actually enlighten you how multiple different data that are connected, collected, high throughput sequencing data and omics data are collected, and how these data are processed, and how very many meaningful biological and diagnostic insights can be developed from these data. So you can follow the gradual steps from collecting the data from the samples, and step by step how to convert the raw data, which is completely unstructured as we get from many of the experimental techniques, and then convert those unstructured data into structured data, and then how to do necessary pre-processing and statistical methods on those structured data to derive very many meaningful biological insights. That's our ultimate aim, to derive many meaningful biological insights. So our question, developing a biological question, is going to be based on this: how can I understand the biological system? So in that sense, we can, and following this workshop, and you will be more interested in like something like mutation analysis that Dr. Harpreet is going to talk about in the workshop, and something like transcriptomic analysis, which can multiomics analysis, which can be used to identify cancer subtypes that will help in precision medicine. You are more interested in this. This directly relates to diagnostic tools that one can relate with. So so you are also working in translational biology, so you can find out how bioinformatics is going to enable translational biology biological insights. So that is what this workshop is all about, and we will demonstrate you, as I mentioned, we will demonstrate you multiple different tutorials and lessons and courses and programs. They are designed in such a way that you can follow through the lessons if you can understand the logic. So I think at this age of our scientific career, every one of us is going to be very well versed in logic. You don't have to have a prior introduction to coding. If you have prior introduction and prior expertise in coding, then we have lessons that cater to you too, by extending our same logical analysis to Python and also to R lessons. So if you do not have if you do not have prior experience in coding or prior experience in bioinformatics, you are also very much welcome to come through to attend the workshop and listen to us and interact with us. The workshop and also the programs are designed for people who are not an expert in bioinformatics. So I think that answered your question. And also there was one more from Paloma, I think, mentioned about not knowing bioinformatics much at all. So you don't, there is no such requirement to be an expert or to be to develop an awareness in, to have had an awareness in bioinformatics. Okay, yeah. I hand it over back to Sonalika regarding the time zone and other certification questions that are popping up.
Thank you so much, Dr. Raghavendran. And this is in the IST time zone. So I think it will be best suitable for Asian countries and African countries and Australian countries. So anyone in this particular time zone can join the workshop, and others can get the recording of the workshop. So if you are in CST time zone and it's not suitable for you to be a part of the workshop, then you can definitely go to the recording. And for the certification part, this is the certificate that you will be getting on completion of the 3 days workshop on Introduction to Computational and Biomedical Data Science. And any other questions? Okay, so I think that we are done with today's session. If you have any question and if you face any issues while enrolling yourself for the program, please feel free to mail us at marketing@omicslogic.com. And Miss Parshthar will be there to help you out with your enrollment. And yes, that was it for today's webinar. It was very nice to have you all for the webinar, and we are looking forward to meet you all in the workshop and brainstorm together. Thank you so much, everyone, and the mentors for being a part of the webinar.